In this video, I will look at the main parts of light microscopes, consider some of the principles of physics involved in microscopy, and discuss some functions of a microscope's controls. There are many types of light microscopes. Most of the microscopes used in student labs have these basic parts: the base with a built-in light source, and an arm. You should carry the microscope with two hands, one on the base and one on the arm. The arm supports the head and a revolving nose piece that holds objective lenses. In this case, a scanning objective barely visible in the back, a low-power objective, a high-power objective, and an oil immersion objective. The nose piece revolves to move each objective into place as needed. On the head are oculars, the lenses through which we see the magnified images. Below the objectives is a stage where a slide is placed, and below that is a condenser that concentrates light onto the slide, and a diaphragm that changes the diameter of the light beam. The position of a slide on the stage is controlled by mechanical stage controls, here barely visible in the back. On the arm are controls for focusing, coarse and fine adjustments. We use coarse adjustment with scanning and low-power objectives, and we use fine adjustment with high power and oil immersion objectives. Light is electromagnetic radiation, that is, energy that travels through space in the form of waves. Types of electromagnetic radiation are distinguished by their wavelengths, the distance from one point on a wave to the corresponding point on the next wave. We name electromagnetic radiation based on its wavelength. Shorter wavelengths are gamma rays, X-rays, and ultraviolet light. Longer wavelengths are radio and TV, microwaves, and infrared. Our eyes respond to electromagnetic waves that we call visible light. We perceive wavelengths as color, from violet, which has a wavelength of about 400 nanometers, to red, which has a wavelength of about 700 nanometers. The lenses inside the objectives and oculars of a microscope bend light to produce magnification. Let's examine how this works. Light travels at a constant speed through a material, but it can change speed when it moves into a new material. For example, light travels in the air faster than it travels through glass. If a beam of light strikes a piece of glass straight on, the entire width of the beam slows at the same time, and we don't see any difference. But if light strikes at an angle to the glass, one side of the beam slows before the other side, and the light bends as it passes into the glass. When it leaves the glass into the air, the process reverses. This bending, which is called refraction, is the basis of magnification. In a microscope, the surface of a lens is at an angle to the path of the light, so the light refracts. The result of refraction is that the light rays from the two sides of an object end up further apart than they were originally. We perceive the new positions of the light rays as magnification. Notice that because the light rays cross one another, a microscopic image is not only magnified, it is also upside down and backwards. An important concept in microscopy is resolution. Resolution is the ability to distinguish between two objects that are close to one another. For example, inside this circle you can resolve two dots, particularly when the image is magnified. When resolution is poor, magnification makes a small blurry image into a big blurry image. It's empty magnification. Resolution depends on a number of things, including the wavelength of light. Shorter is better for reasons we won't go into, the angle of the surface of the lens, and the difference in the speed of light in the lens from the speed in the air. Now, let's consider the use of oil to improve resolution. When light passes through a glass slide and a specimen and back into the air, it refracts and scatters. Some of the scattered light doesn't enter the objective. At lower magnification, this loss is negligible, but at the highest magnification, scattering matters. Immersion oil has exactly the same optical density as glass. That is, the speed of light in glass is the same as the speed of light in immersion oil. Therefore, when oil is between the slide and the oil immersion objective, the light doesn't scatter. All the light enters the objective, and this improves resolution. In summary, we reviewed the main parts of a microscope and their functions, saw that lenses magnify by refracting light, and considered the use of immersion oil to enhance resolution.